Hello, this is Dr. Neela Bakore, and we have started with the chapter of photosynthesis, and we discussed some basic things, some historical things, plus the types of pigments which are required, structure of chlorophyll, the various types of chlorophylls and other pigments, and we also saw the light quality that is essential for photosynthesis along with action and absorption spectrum. Now, let us see this photosystem, the actual collection of pigments which are helping or which are going to help in initiation of this process of photosynthesis. Photosystems. There are two photosystems and they are written as PS1. PS stands for photosystem. So, PS1 and PS2. We will take these systems in details in few minutes, but let us talk about the scientist and how it led to the discovery that there are two photosystems. The scientist is Emerson. So, Emerson was trying to expose plants to various wavelengths separately. So, from the complete visible spectrum, so from 300 nanometers to 700 nanometers, and he was trying to count the quantum of photosynthesis, and it was done on the basis of emission of oxygen. So, the more the oxygen released would indicate that there is more photosynthesis. So, amount of oxygen released per quantum of light absorbed. This was the criteria to find out what and how much of photosynthesis takes place at different wavelengths. So, when it started with 300 nanometers, it went up to 700 nanometers. He observed that the photosynthetic graph, there was a sudden drop in the rate of photosynthesis after red wavelength. So, when this graph was plotted, he observed that after the wavelength of red, which is somewhere around 780 to 700 nanometers, sorry 680 to 700, there was a sudden drop in the rate of photosynthesis. It was called red drop. Then he observed the same rate of photosynthesis by combination of wavelengths. So, he exposed the plants to a wavelength of 680, before that 650, approximately 650 plus something, and then 700 nanometers separately. So, he found that around 680, the rate of photosynthesis was, suppose we just represent it in the form of a small bar chart. This was at 650, 680 only. When the plants were exposed, the rate of photosynthesis was slightly similar or slightly higher than that at 700 nanometers, when these wavelengths were given separately or when the plants were exposed to these wavelengths alternatively. So, once 680 approximately, then 700. So, when these two wavelengths were given, this is how the rate of photosynthesis was. So, it was pretty much same. Then he exposed the plants to combination 650 plus 700 together, and it was observed that the rate of photosynthesis increased. So, this led to the discovery of photosystem 1 and 2. It indicated that there are two individual units which are helping in photosynthesis. One unit works at around 680 nanometers and the other unit works at around 700. So, when both the wavelengths were given simultaneously, both these units were working together, and that is why the rate of photosynthesis was higher. This was called Emerson's effect, or it was also called Emerson's enhancement. So this experiment led to discovery of photosystems and photosystem 1 and 2, the two units that exist and which help the plant in photosynthesis. Now let us talk about what exactly is this photosystem. Originally it was called photosynthetic unit or quantasome. Nowadays we call it photosystem 1 and 2. So it can be written as photosynthetic unit or quantasome, but the new term which we use is photosystem. Let us first see what exactly is a photosystem, and then we will see what is the difference between system 1 and 2. A photosystem is a collection of pigments. So there are pigments. Which pigments are there in this system? Mainly chlorophyll. So there can be chlorophyll, carotene or carotenoids, xanthophyll and phycobilins. So we have talked about all these pigments. So it is a collection of pigments. There can be any type of pigment. The approximate number is about 230 to 240 chlorophyll molecules are there. So pigments are present. Plus there is an electron acceptor. So if we make this collection of pigments, suppose we are drawing these pigments, this collection of pigments is also known as antenna complex. So this is antenna complex, which is nothing but a collection of pigments. Here one pigment is the most important one, and this important pigment is known as reaction center. This reaction center pigment is always chlorophyll A. So whether it is PS1 or PS2, the reaction center chlorophyll is going to be always chlorophyll A. In PS1, this chlorophyll A absorbs 700 nanometers, the wavelength which is absorbed by the chlorophyll pigment. If it is 700 nanometers, then this photosystem will become photosystem 1. It is written as P700. If it absorbs around 680 nanometer wavelength, then that reaction center containing photosystem will be PS2, and it is written as P680. So the difference between the two is the reaction center. Chlorophyll A, if it absorbs 700, it is PS1. If it absorbs 680, then it is PS2. For our understanding, we are drawing the electron acceptor slightly away from it, but it is a cluster in which there are pigments also and this electron acceptor also. Just to understand it, we are drawing it separately. This is the primary electron acceptor. It is going to accept the electrons. The electron is going to come from chlorophyll A. We will see that in a minute. So what exactly are the things which are present in a photosystem? In a photosystem, there is a cluster of pigments which is known as antenna complex. In this cluster, there is one main chlorophyll which is called reaction center or reaction center chlorophyll, which is always chlorophyll A. If it absorbs 700 nanometers, it is a part of photosystem 1. If it absorbs 680, it will make photosystem 2. Each antenna complex will have its own primary electron acceptor. So, when the electron jumps from here, it will be accepted by this electron acceptor. Why are we calling it primary? Because it is the first one to accept the electrons. We will see what different types are there. Now, this is this chlorophyll A, which is the reaction center chlorophyll. This complete unit is photosystem or quantosome or photosynthetic unit. Photosynthetic unit, as we said, it is an older name. Nowadays, photosystem is the more commonly used term. This main chlorophyll, which is the reaction center chlorophyll, will start the reaction or will start the photosynthetic activities. Now, this primary electron acceptors are different for PS1 and PS2. So, this is the chlorophyll A, which absorbs 700 nanometers in PS1 and 680 nanometers in PS2, and the primary electron acceptor. In case of PS1, this primary electron acceptor, there are two schools of thoughts, rather we can say there are two groups of scientists. One group believes that it is an unknown substance. The other group says that it is an iron sulfur containing protein. So, the primary electron acceptor in case of PS1 is an iron sulfur containing protein, but other scientists feel that it is some molecule about which the information is not available, or we do not know exactly what is the nature of that one. In case of PS2, there are again two schools of thoughts. One group says that it is a chlorophyll A molecule which is colorless. So, it is a colorless chlorophyll molecule in which the magnesium ion which is present in the head of chlorophyll, in the porphyrin head, when we did the structure, there is magnesium in the middle and then there are 4 pyrrole rings around it. So, that middle magnesium is missing. So, it is a colorless and the magnesium ion is absent. So, some scientists believe that primary electron acceptor in PS2 is a colorless chlorophyll molecule in which in the porphyrin head there is no magnesium core. Other scientists believe that it is a substance called quinone. So, we will take both, we will write either quinone or this. Quinone is written as capital Q. So, these are the two differences between PS1 and PS2. One more difference is the pigments which are present in this cluster. PS1 has more of keratinoids. So, the pigments which are there other than this main, in case of PS1, it has chlorophyll A, chlorophyll B plus mainly keratinoids. The ratio is about 20:1. So, normally there are 200 to 400 chlorophyll molecule and maybe 1 or 2 keratinoids, but these two main pigments. In case of PS2, along with chlorophyll, the other pigments which are present are phycobilins. So, this is pigment wise difference. So, PS1, PS2 differ in 3 main things. The reaction center chlorophyll A, the wavelength which it absorbs, 700 in PS1, 680 in PS2. The electron acceptor wise, PS1 has iron sulfur containing protein, again some scientists believe like that, and in PS2 there are 2 things which probably are the electron acceptor. One is a colorless chlorophyll molecule without magnesium, and the other is quinone, represented by Q. The third difference is in the pigments which are present. The pigments other than the reaction center chlorophyll, in case of PS1, chlorophyll A and B are present and mainly carotenoids, whereas in case of PS2, along with chlorophyll, plus phycobilins are more important. Now, what is the role of these other pigments? This main is the reaction center. We said reaction is going to start from here. What are these other pigments going to do? These pigments are called accessory. Accessory pigments. Accessory pigments means helping pigments. We will see how they help in this photosynthetic process. So, reaction will always start with reaction center chlorophyll. Accessory pigments, it can be A, B, all other categories, they are going to help. And when electrons are emitted by chlorophyll, those electrons will be accepted first by an acceptor. This acceptor is known as the primary electron acceptor. Now, as we have seen the differences between photosystem 1 and 2, and also the scientist who discovered that there are two photosystems, two important things which we have talked of, red drop and Emerson's effect, very, very important whenever it comes to photosystem and its discussion. We will talk about the two phases in which photosynthesis is completed, the light reaction and the dark reaction, and simultaneously we will take up few more interesting things. Now, before we take up light and dark reaction, we need to understand one more important thing, that is fluorescence and phosphorescence. The difference between the two, if chlorophyll is extracted, we won't see these things in the leaf or in the cell which is a part of the plant. So, if chlorophyll is extracted, suppose this chlorophyll is extracted, that means isolated from the plant, say it is in a test tube. Then if light falls on this chlorophyll, again photon or the solar energy, so this light which falls on chlorophyll, it will excite a pair of electrons. So, now in this chlorophyll, suppose we talk about this chlorophyll and it has its shell, and here there are two electrons. So, when light falls on this chlorophyll, on these electrons, they absorb these photons and they get excited. The amount of energy increases and they get excited. So, from this original ground state, these electrons move to the higher level. So, from the ground state, the electrons go to excited state. So, now from this place, the pair of electron has jumped to this place. Normally, it is the tendency of every particle to remain in its lowermost energy state. So, this electron would tend to come back to its ground level. Here we need to understand, why did the electron jump? The electron jumped because it absorbed energy. So, its energy increased and that is why it jumped from the ground level to the excited state. It has a tendency to come back. So, when it comes back from the higher energy level to the lower energy level, what is going to happen to that extra energy? That extra energy is emitted and it is seen in the form, it is a wavelength again. So, depending upon which wavelength is emitted, that is the color which we are going to see. So, there is a flash of wavelength which is given out, and this is seen in the form of fluorescence. So, there is one flash of light or wavelength which is given out and we see it as fluorescence. Sometimes this electron, when it is coming back, this is the higher level, when it is coming back, it gets trapped in a band here. This is a transition band. Now, if I magnify this, suppose this is the higher energy level, this was the lower energy level. If the electron pair comes back, or let me use another color, when it comes back to its ground state, it gives away some wavelength. This is fluorescence. But if this electron pair, when it is coming down, it gets trapped in a transition band. In this transition band, it is called electron spin resonance, in which the electron keeps moving between this band. So, it remains trapped here, and as it remains trapped here and it is moving in this band, it keeps emitting energy, again wavelength, for a longer period of time. This is known as phosphorescence. So, chlorophyll shows both these features, that is fluorescence and phosphorescence. Condition is it should be extracted. We would not see any of these two things if it is in the plant, because in the plant what is happening is when from chlorophyll A the electron pair gets to the higher level, it is accepted by the acceptor here. Whereas in this case it is an experimental condition outside the living system, that is outside the cell or outside the plant. There is no primary electron acceptor, we have simply isolated chlorophyll. So, when light falls on chlorophyll, a pair of electron gets excited, it goes to the higher level. So, from ground state it goes to excited state. In normal condition it would come back in one go, and that extra energy would be released in the form of some wavelength, that is known as fluorescence. Sometimes the electron which is, or electron pair which is coming down, gets trapped in a transition band, and there is electron spin resonance, it keeps moving in that band, and because it is in this band, it keeps emitting wavelengths for a longer period of time, and that wavelength, or rather those wavelengths which are emitted, are known as phosphorescence. So, fluorescence is for a shorter period of time because it is one step and it comes down to the ground level, whereas phosphorescence takes place for a longer period of time. So, chlorophyll shows both these things in isolated condition, not inside the plant or inside the cell. Inside the cell, the electron will be accepted by the acceptor, electron acceptor. Now, let us talk about the two phases, that is light reaction and dark reaction. We will start with light reaction first.